seed that falls on good earth is a seed to harvest spring. The good earth is full of fungus, bacteria, and other things. I'm here to tell you why the pea plant doesn't mind, so long as you don't plant it in the same place every time. Rotate, 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 rotate. I looked through 200 plants in this greenhouse planting, which you'll see in the next slide. And I found one plant with root rot. If I replant peas in the same soil, I will get a higher number, and if I continue to replant in this soil, the disease will reach epidemic proportions. Now, if I plant peas in a soil that was previously planted to beans, the peas are still happy because of a phenomenon called non-host resistance. That is, the pea plants are non-host to the bean pathogens, and after 20 days, the pea plant totally resists the bean challenge. How do plants, this is what the disease looks like. How do plants resist disease? Plants have PR genes, or pathogenesis-related genes. What do those pathogenesis-related genes do, and why do they become active? These are the pathogenesis-related genes. Some of them are involved with things like lignin synthesis, or digestion of fungal cell walls, or some intermediary pathways result in the production of a small antifungal compound called physotin. Then the ones on the bottom that you see, DRR230 and DRR39, are defenses. They're small peptides that are antifungal directly. These peptides are found throughout the plant and animal kingdom, and they're in frog skins, in your red blood cells, and even in scorpion toxins, so they're very potent chemicals. Dr. Cruikshank in Australia found a way to shorten the cycle that disease resistance takes. In other words, instead of putting it on the fungus on the plant, you put it on the inside of a pea pod, and within 6 hours, the pea pod can resist a bean pathogen. And that is essentially the system that we've worked with for a lot of years to see what the nuclear changes are that occur after the fungus arrives to this tissue. Now, gene activation begins at the nucleosome level. The polymerase complex that transcribes genes moves when the nuclear proteins are temporarily dissociated from the DNA. Here is what a nucleosome looks like. You can see the red and yellow histones that are sticking to the DNA helix that goes around the outside of the picture. Those must be dissociated to allow the polymerase complex to go through. This is all of the things that happen in the host-parasite interaction that we've been able to find. And it's a complicated picture, but I want to just concentrate on the left-hand side. This is the bean pathogen infecting the pea tissue. And there's two signals that come out from the pea fungus. One of them is a DNase, and this has the capability of making single-strand nicks in the DNA of the plant cell. On the other side, there's a compound produced called chitosan. It has an ability to compete with histones that are also attached to the DNA molecule. And when this conformational changes occur in the DNA or the proteins associated with the DNA, there's a PR gene activation. This is just a little summary of what goes on there. The DNA comes down the line, and the polymerase unit comes down the line on the DNA, and the ubiquitin is one of the things that helps dissociate the histones away from the DNA. Down you go a little further, and you see there's a break that can occur with the fungus DNase. And as you go a little further, you can see ubiquitin is also pulling off a transcription factor called high mobility group protein. And then finally, chitosan is able to compete with the histones directly. How do we know that the major nuclear proteins are altered in the vicinity of the PR genes of interest? Well, you can purify these proteins, but you have to have the antisera first. The associated DNA sequence next to the protein can be identified by an assay called chromatin immunoprecipitation. This is what the data looks like on that. If you look at those bands, the first two had to do with the water control. The second two are when the fungal challenge has occurred in the tissue after 4 hours, 5 hours. So it is possible to say that the proteins that you're dealing with are being changed at the level of your gene that you're interested in. And the 206 is one of the PR genes that we talked about. How do we know that nuclear proteins are being dislodged by ubiquitination? Well, the nuclear proteins get bigger because as they complex with ubiquitin, they move slower in a gel, so you can detect them. And that is a covalent linkage, so they stay together on the gel. This is a picture of ubiquitinized proteins on a gel, and they run far above the area where the unubiquitinized protein would migrate. Thus, the pea plant is a non-host to the bean pathogen. The bean pathogen unwittingly releases components that, such as the DNase, that can alter the nucleosome structure of the pea in the region of these PR genes. And then when these genes become active, the protein products and their subproducts, like the small phycetin molecule, return to the fungus with their antifungal action. The following scheme is a prediction from computerized determinations taking just the N-terminal sequences of the gene products that we've talked about. You can see here that anytime that arrow goes from the fungal spore into the plant, the computer says it can get out of the fungal spore cell and get into the plant cell. And we know that the DNase does get into the nucleus of the plant, and it does cause single-strand nicking there. As a result of these conformational changes, the PR genes are induced, and some of them are also capable of getting out of the cell. As you can see, DRR39, DRR230, and DRR206, as well as the two cell wall degrading enzyme products do get out of the cell. More details of this disease resistance can be found in Phytopathology 98-372, 2008 publication. Thank you very much. Did I use all my time? I probably did.
I don't know.